Welcome to our video. We are doing the electron transport choo-choo train.
And ATP synthase too.
So to understand the electron transport chain, we need to have an overview of cellular respiration. And so the first step of cellular respiration is glycolysis, which takes place in the cytoplasm. And then that leads into the citric acid cycle, or the Krebs cycle, which takes place in the mitochondria. And then that leads to the electron transport chain, which is the last step, which also takes place in the mitochondria. So here are some pointers that go over what we just talked about if you want to pause and review.
One common misconception of the electron transport chain, or the ETC, is that its purpose is to create the energy, which is not true. The purpose of the ETC is to release the energy in smaller, manageable amounts instead of all at once.
Okay, so now we're going to discuss electron transporters. The two main electron transporters are NAD+ and FAD. And their purpose is pretty much what it sounds like. They shuffle electrons back and forth to keep the process of cellular respiration running. When these compounds pick up electrons, or in other words, when these compounds are reduced, they become NADH and FADH2. It's kind of confusing because in a reduction reaction, you actually gain electrons, so that's what's happening here. And these reduction reactions take place in the citric acid cycle, which takes place in the mitochondrial matrix. And the electron transporters head to the electron transport chain on the mitochondrial intermembrane. NADH and FADH2 drop off their electrons and they go back to their original state, or they are oxidized into NAD+ and FAD. Because remember, an oxidation reaction means losing electrons. And then they just go back to picking up more electrons.
All right, so we are going to talk about the hydrogen gradient, which is also called the proton gradient, and it's a difference in concentration between the inner and outer membrane of the mitochondria. So in the previous slide, NADH and FADH2 have just dropped off their hydrogens, and so now the hydrogen is in the gradient. And so this is going to give the actual push into the production of the ATP synthase. So this is super important. Without this gradient, ATP could not be formed in this manner. All right, now we get to talk about ATP synthase, and this is where the magic of ATP is formed. So hydrogen is like this little token, and it goes down the little chute, and it sees ADP and an inorganic phosphate, and it's like, guys, you guys should totally hang out. And they're like, yeah, we totally should. So then they get together, and that is how ATP is formed. And this is just a glorious thing, and ATP is just so beautiful because without it, we would not really survive.
So now you're going to ask yourself, what about the hydrogen ion? Because it's just created this beautiful coupling of ATP, but it's quite lonely and sad. So hydrogen ion is going to exit the ATP synthase, and it's going to hook up with its best friend, an oxygen molecule, and it's going to create water. And the reason that this is important is because if the hydrogen ions stay single on the other side of this membrane, the gradient is going to become more equilibrium, which makes the other side of it, with all the hydrogen ions, if it's evenly split, it's not going to have this draw to go down the ATP synthase to correct the gradient. It will equal out. So hydrogen hooks up with oxygen. It leaves the system through the matrix of the mitochondria, where it will either be used as the cell, used in the cell, in the mitochondria, or wherever it's needed in the whole system. Not all of these hydrogen atoms are going to hook up with oxygen. Some of them will go back and find the FAD plus or NAD plus and go through the system all over again. And that's how we get our choo-choo train to continue to go on and on and on throughout that whole thing.